Hello everyone, welcome to Sapimia Tutorials. Today we are going to continue with the inflammation. And before I continue, I want you to hit on the subscribe button to subscribe to this channel. You can also write something in the comment section, and you can also share this video to your friends. Alright, so let's continue. In our previous video, we've been talking about inflammation, and we've looked at the initiation phase, the vascular phase, the cellular phase, the resolution phase. So this is just a brief summary, or this is just an overview of what we've done so far at the inflammation. So we talked about the initiation phase. So under the initiation phase, that is where we talk about the pathogens being recognized by immune cells and leading to the activation of immunological activities. That is when the pathogen-associated molecular patterns binds to the pattern recognition receptors to initiate or to trigger a series of immunological activities or immunological events. So we went on to look at the various chemical mediators that are released, including vasoactive amines, lipid mediators, chemokines, pro-inflammatory cytokines, etc. So these chemical mediators results in the vascular and then some processes in the cellular phase. So we talk about the 5 cardinal signs of inflammation: vasodilation, vasopermeability, etc. And we talked about the cellular event. So that was where we ended. Under the cellular event, we talked about leukocyte extravasation. That is the movement of leukocytes from the vascular compartment into the interstitial space to undergo phagocytosis or to attack pathogens. Today we are going to look at phagocytosis. So right now the leukocytes or the immune cell is now out of the vascular compartment and is now in the interstitial space. What is going to happen next? So the next event is what we call phagocytosis. So today we are going to look at phagocytosis. All right. So there are basically 4 main stages under phagocytosis. So the first one is the recognition and attachment. The second one is engulfment or ingestion. The third one is Phagolysosome Formation. And the fourth one is destruction and digestion. So let's take a look at these 4 main processes under phagocytosis. So let's begin with the recognition. Okay. So we already know that immune cells have structures on them we call pattern, or receptors on them we call pattern recognition receptors. And pathogens also have pathogen-associated molecular patterns. So this is one of the ways the immune cells are able to recognize the pathogens, through their pathogen-associated molecular pattern. So once they identify the pathogen-associated molecular pattern, they go to them, they bind to them, they initiate their engulfing properties, and then they finally engulf and digest the pathogen. So the first way of recognizing the pathogen is through the PRR-PAMPS interaction. That is pattern recognition receptor and pathogen-associated molecular pattern interaction. Now the second way immune cells or WBCs are able to identify pathogens and attack them is through opsonization. So now you can see here that we have antibodies and then we have complement proteins. Now let's begin with antibodies. So when antibodies surround the immune cells, what they do is that they synthesize them or sensitize. Yeah, that's the right word. They sensitize them for engulfment. So the moment that is what we call opsonization. And it's not only the antibodies that is responsible for the opsonization, the complement proteins as well. So when we do complement system, you realize that some of the complement protein act as opsonizers or opsonization agents by what? Opsonizing the pathogen, making it so sweet, so delicious for the pathogen, for the immune cells to engulf. So that is the way that these are the 2 main ways the immune cells or the white blood cells are able to recognize the pathogens and engulf them. Now let's look at the second stage. The second stage is the engulfment stage or the ingestion stage. So this is a pretty simple stage. So now let's talk about this one first. You could see macrophages, dendritic cells, and the neutrophils. These are the three major cells that are involved in phagocytosis. These are the three main cells that are involved in phagocytosis. So in the engulfment stage, what happens is that the immune cell, or the WBC, after it has recognized the pathogen, it surrounds its membrane by extending its membrane around the pathogen. So you could see there's an extension of the cell membrane of the immune cell around the pathogen. So the immune cells want to engulf the pathogen, but first of all, it have to surround its cell membrane, extend its cell membrane to surround the pathogen. Now, this is the next process. You could see here that there is formation of phagosome. There is formation of phagosome. So the main purpose of the engulfment is to form a phagosome. So right now you could see that the membrane has finally extended. It's in the process of forming the phagosome, and here you can see that our phagosome is already formed. So you could see there is a membrane around the pathogen, and this membrane is what we call the phagosome. So now the cytoskeleton and everything of the membrane will now readjust to itself. So it now assume its normal shape. After the extension has been done. So now we can see our phagosome here, and then we can see our pathogen right in our phagosome. So that is the first stage. That is the engulfment stage. So the cell membrane of the WBC throws itself around the pathogen. It circles it or engulfs it to form what we call phagosome. So that is the engulfment or the ingestion stage. Now, our next stage is going to be the phagolysosome formation. So as you can see, the word phagolysosome, we can derive two words from it. The word phagolysosome, we can derive two words from it. We can have phagosome and lysosome. So the main purpose of this stage is to fuse the lysosome and the phagosome together to form what we call a phagolysosome. So this is what happened. So every cell has a lysosome. The function of a lysosome in every cell is to initiate autolysis or autophagy. So that is self-destruction or destruction of worn-out tissues or pathogens. So lysosome contains hydrolytic enzymes, and these hydrolytic enzymes helps in the digestion of pathogens and worn-out tissues or worn-out cells. So that is the function of the lysosome. So now we have in our cell the phagosome and then the lysosome. So let's see what happens next. So what happens is that the lysosome begin to join its membrane to the phagosome to fuse and form what we call the phagolysosome. So we realize that there is a formation of one particular membrane or one particular entity. So that entity is what we call the phagolysosome. So you can say phagosome and lysosome fusion. Okay, so after the fusion is done, we will now have our phagolysosome here. So now you can see our hydrolytic enzymes here, and then you can see our pathogen here. So this is what we call the phagolysosome. All right. So now we are going to our last stage of the phagocytosis. That is the destruction and the digestion of the pathogen. So now we have our pathogen here, and then we have our hydrolytic enzymes here. So the hydrolytic enzymes was actually in the lysosome, and then the pathogen was actually in the phagosome before fusion. So now after fusion, all of them comes together to form one entity we call phagolysosome. Okay. So now how are the pathogens, or how is the pathogen destroyed? So we have two ways in which the pathogen is destroyed after a phagolysosome fusion or after a phagolysosome formation. So one, the hydrolytic enzymes that are present within the lysosome attacks and digest the pathogens. So that is one way the pathogen can get destroyed. The hydrolytic enzymes that are found in the phagolysosome attacks the pathogen and then digests it. Now, the next thing that happens, or another way in which the pathogen can be destroyed, is through reactive oxygen species formation. So now what happens is that the membrane of the phagolysosome, and I'll go through a series of chemical reactions to produce reactive oxidant species. And these reactive oxidant species could be superoxide dismutase. It could be hydrogen peroxide. So those oxidative species, it could be nitric oxide. These oxidative species, and that goes or attacks the pathogen through what we call oxidative burst. So oxidative burst will destroy or eliminate the pathogen. So these are the two ways the pathogens are destroyed after a phagolysosome fusion. Okay, now what happens is that after this destruction is done, the fragments, or what we call the antigens that are released, move into the lymphatic system through the lymphatic vessels, and then continues with the adaptive immunity. So you could see antigen move to the lymphatic system to continue the adaptive immunity. So we can say that this becomes the bridge. This stage becomes the bridge between the adaptive immune system and the innate immune system. Alright, so this is what phagocytosis is all about. In our next video, we are going to talk about the complement system, which is also one of the types of innate immunity. We are going to talk about the complement system and how it contributes to the innate immunity. Before you go, I would like you to hit on the subscribe button to subscribe to this channel and get more medical insights simplified. Alright, thank you very much and see you next time.